Discovery of the structure of DNA. This video was created for Ohio Valley University's NSC 432 genetics class in the spring of 2012 under the instruction of Mr. Gordon Wells.
By the late 1940s and early 1950s, it was becoming widely accepted that deoxyribonucleic acid, DNA, was the genetic material. Once this was shown through various experiments by Griffith, Avery, Hershey, and Chase, there was a big push to understand the physical structure of DNA and how its structure was related to its function as the genetic code.
Many scientists were working feverishly towards understanding the structure and function of DNA. Some of the main scientists involved in this race were Linus Pauling, Maurice Wilkins, Rosalind Franklin, Erwin Chargaff, James Watson, and Francis Crick. We have ordered this video in a way that should allow you to see how the various information was compiled. All of these scientists were working at the same time and made contributions to the discovery of the structure of DNA.
In the 1950s, Linus Pauling made a proposal about the folding of proteins into an alpha helix. He modeled this structure using ball and stick units. Most of his studies revolved around proteins. Like many scientists of the time, he was still stuck on the notion that proteins were the genetic material. Most things that he wrote about nucleic acids were in the thought line of nucleoproteins rather than the nucleic acids. Looking back, many were astonished that Pauling did not discover the structure of DNA on his own. This was probably due to his fixation with proteins. Looking back, he thinks that part of the problem was that he didn't feel the need to work or collaborate with anyone else. Even though Pauling was not the one to discover the structure of genetic material, his work on the structure of proteins and his methods of modeling were adapted by James Watson and Francis Crick, who will be discussed later on.
Wilkins obtained a Ph.D. in 1940 with a thesis dealing with thermal stability of trapped electrons in phosphorus and the theory of phosphorescence. He applied these ideas to different wartime efforts. He also worked on the Manhattan Project. Once the war was over, he lectured on physics at a Scotland university. Here, he began to research biophysics with John Randall. When the biophysics study moved to England, Wilkins did also. Here he studied the nucleic acids in the tobacco mosaic virus. He focused on the orientations of purines and pyrimidines. He then began working on X-ray diffraction studies of DNA and sperm heads. Wilkins had begun to implement the study of DNA by X-ray crystallographic techniques when Rosalind Franklin was appointed as a research associate by Randall. The two did not have a good relationship. Wilkins shared Franklin's famous X-ray of DNA with James Watson without her permission.
Rosalind Franklin is perhaps the woman scientist with the most controversy surrounding her life and work. Born in 1920, she decided at the age of 15 that she wanted to become a scientist. In 1941, she graduated from Newnham College, and in 1945, she graduated from Cambridge with a doctorate in physical chemistry. From the time she graduated until 1950, she worked in Paris, where she learned and soon mastered X-ray crystallographic diffraction techniques. In 1951, she moved to England to work as a research associate in John Randall's lab. This is where she met Maurice Wilkins, who worked in the same lab and mistakenly took Franklin as a technical assistant rather than his peer. They worked on separate projects that both involved DNA. Between 1951 and 1953, Franklin became very close to solving the structure of DNA. Her famous crystallographic portrait of DNA was a major factor in discovering the structure of DNA. To obtain such a clear X-ray, Franklin adjusted her equipment so that it produced an extremely fine beam of X-rays and extracted finer DNA strands than ever before and arranged them into parallel bundles. This X-ray was critical in determining the structure of DNA.
Erwin Chargaff was born in Austria in 1905 and obtained a doctorate in chemistry from the University of Vienna. His most important contribution to science was his work with DNA and nucleic acid bases. The reason that his name is so familiar is that Erwin Chargaff has a set of rules named after him. These rules are important to the structure and function of DNA. DNA consists of sugar, deoxyribose, phosphate, and bases. The bases are adenine, thymine, guanine, and cytosine. Through various experiments, Chargaff noted an important relationship between the nucleic acid residues. The number of adenine residues always equals the number of thymine residues, and the number of guanine always equals the number of cytosine. Since A and G are purines and C and T are pyrimidines, the number of purines always equals the number of pyrimidines. These rules were important in discovering the bonding within the DNA molecule. From this information, it is possible to conclude that A must bond with T and C must bond with G. The double helix of DNA might also have been derived from this information, considering the idea that they are paired with each other.
James Watson, born in 1928, was an American-born scientist who had a degree in zoology and a strong desire to learn more about genetics. This driving interest in genetics eventually led him to the zoological station in Naples in the spring of 1951, where he met Maurice Wilkins. Wilkins gave Watson his first view of the X-ray diffraction pattern of crystalline DNA. This prompted Watson to change his direction of research towards the structural chemistry of nucleic acids and proteins. Luckily, he was able to pursue this through an arrangement with John Kendrew to work at the Cavendish Laboratory at University of Cambridge. He started work there in October 1951, where he soon met Francis Crick.
Francis Crick was born in England in 1961. In 1937, he received a Bachelor of Science degree in physics from University College London. He was a member of the Medical Research Council unit, which was housed for many years in the Cavendish Laboratory. Here he met and formed a friendship with James Watson in 1951. He gained a Ph.D. in 1954 on a thesis about X-ray diffraction of polypeptides and proteins.
Together, Watson and Crick made their first serious effort in determining the structure of nucleic acids in 1951. This effort was less than satisfactory. Their second effort, in which they gathered much more information from others, proved to be more fruitful. Integrating the information they gathered from the work of others, Watson and Crick were able to propose a double helical structure of DNA along with the pairing of bases on the inside of the helix. They proposed this structure in 1953 when it was published in Nature.
Their famous paper, titled "Molecular Structure of Nucleic Acids," was only a short article, barely a page and a half long. At first their article did not gather much attention, presumably due to the title. However, in years to come, their work would be considered one of the greatest discoveries in science.
For their efforts and contributions, several scientists were recognized. Linus Pauling received a Nobel Prize in Chemistry in 1954 for his work on the structure of proteins. Watson, Crick, and Wilkins received a Nobel Prize in Physiology or Medicine in 1962 for their work on the structure of DNA.
Although Watson and Crick are credited with the discovery of the structure of DNA, they did very little of the actual work themselves. They considered the work of Linus Pauling and mimicked his modeling system of ball and stick units, as well as keeping in mind the helical structure of proteins that he suggested. Watson was also fortunate enough to use his friendship with Wilkins to get a view of Franklin's excellent DNA X-ray crystallography portrait, even without her permission. He then related this information to his colleague Crick, who was also a high-caliber X-ray diffraction scientist. This information of a helical model and X-ray pattern was combined with the work of Erwin Chargaff, who noted the relationships between the nucleic bases. This allowed them to piece together the structure of DNA. Franklin died at the young age of 37 from cancer in 1958. Since Nobel Prizes are not given post-mortem, she did not receive one. However, she still made many valuable contributions to determining the structure of DNA. Chargaff, who also made several valuable contributions, did not receive a Nobel Prize and was highly upset that Watson, Crick, and Wilkins did not give him his due credit.
We hope you all found this video informative and helpful. Feel free to leave any comments or post any questions. Thanks for watching.